Welcome, and thank you for having me here. I am the outliers, I guess, from all the speakers until now. I am a basic scientist. We have really, the morning session is a great evidence for the greatness of Yale. This is really a very difficult act to follow for me. So what I would like to concentrate is not on all basic research, as Mario said, that I'll talk about, but some aspects of more recent basic research that is really emerging in melanomas and other cancers. So from my point of view, which I have been in Yale since 1973, participating in melanoma board meeting for the past at least 20, 25 years, and until the past 5 years, I would say completely bored. Steve Arian actually is the one that pulled me. You know, we met in the corridors of Yale and said, Ruth, you have to participate, you have to come. And say, but I persisted, and now it's actually becoming exciting. And the exciting thing is to hear Steve Arian talking about genes. So, in a bird's view, until about very, very recently, all what I could hear about are very toxic compounds like the carbazine and other things. And more recently, Mario and obviously other clinicians brings in the immunotherapy that is really doing wonderful things to melanoma patients, and we have several patients here that walk around probably because of that. But more recently, the word targeted therapy is emerging. And what targeted therapy means? Targeted therapy means, as far as genes are concerned, is to hit the genes that makes the cell work, make the cells proliferate. And right now in melanoma, as you already heard, essentially there are 3 genes that what we call driver gene, and I'll explain a little bit in a minute, that are responsible for continuous proliferation of the melanoma cells or other cancer cells in the tumor. Those genes, the major one is BRAF, which is about 40% or 50% of cutaneous melanoma, NRAS, which is about 15%, and then KIT, which is really restricted to a subgroup or subtype of melanoma, which is acral and mucosal. Now, BRAF kinase is really a target, as was mentioned before, and so is KIT, and we start to see wonderful results with that. So the word BRAF and KIT and NRAS might be right now like a common terms among patients and among scientists, but just to give you a little bit of a timeline and perspective, how they got to be there to begin with. It didn't happen yesterday, it didn't happen 5 years ago. It actually started with 100 years ago with the discovery that there are chromosomes alteration that are involved in cancer. As Daniel, I'll show you some photographs of chromosomes so you get an idea what it is. So this is almost 100 years ago. And then an evidence for that came about 40 years later with the Philadelphia chromosome that I'm sure you have heard about. And another important concept is the 2-hit hypothesis of Knudsen. So Daniel described the inherited genes, and you might think, oh my God, I have an inherited gene, so I'm going to get melanoma. Well, that's absolutely not the case. You have the inherited gene, and this inherited gene is actually also common in individuals that don't have any cancer, exactly the same gene at quite good frequency. So you need to have another hit. And usually the other hit is what you call somatic. It's within the tumor cells. And the 2 of them are the starting of malignancy, but not all by themselves either. Okay, so that's a 2-hit hypothesis. And then the Nobel Prize was given to Bishop and Vermus for discovering the proto-oncogenes that originally started with viruses. And this is 1976, okay, many years ago. And the proto-oncogene, which means genes that can cause cancer, but they are the proto. They are just the normal type that may cause cancer when changed. So the change came with the discovery of HRAS by Bob Weinberg, and this is 1982. So this is now 30 years ago. And since then, I sort of indicated in red, we have 15 oncogenes discovered in 1985. The RB tumor suppressor, which is, by the way, the second hit that is required, is the release of constraint, was discovered in about 1986. Then we have more genes coming up, 120, 200, and so on. So the BRAF V600 mutation in melanoma was published in 2004. And the drug that hit this tumor appeared in 2008. This is 4 years. It's a remarkable short period of time. And the reason it was discovered is that the person who heard the talk about BRAF mutation in melanoma happened to be at Yale. His name is Yossi Schlesinger. And he said, here we have a target. And immediately he had a company at the time, Plexicon, and he said, we have to go after BRAF. And sure enough, 4 years later, the PLX, which is now has other names, obviously, came to the market and is now being treated. And the reason it is so successful is because we are targeting an altered product in the tumor. It's not your normal type. Like Steve said, yeah, normal cells are also affected. They have the same genes. But here we have an altered type that is driving the tumor and more susceptible for this cancer. So where are we standing now? Right now we are standing with 2,300, so to speak, tumor-causing gene or oncogenes. And this is the results of about 190,000 publications, and about 3,000 of them are BRAF. So this is the background of basic research that leads to this kind of discoveries. So right now, if you go to the Sanger site, which are essentially reports of all the genes and mutations that are involved in cancer, the consensus is that 1% of human genes are implicated in cancer. 90% of them are somatic, meaning arising within the tumor and not in your inherited DNA. And 20% of them are inherited, and 10% of them are actually both. They can be in individual inherited and also arise in the tumor. So why do we need so many genes? In a way, it's good, because that means that we don't get cancer really that often. Because the whole process of getting a cancer cell, it's really elaborate and complicated. And I listed some of them. You need the cells to continue to proliferate, to be insensitive to environmental factors that tell them to stop, needs to evade date, to be immortalized, to go forever. Normal cells are not immortalized. You know, I do a lot of cell culture, meaning I take the cells and put them in culture. If I take normal cells, they die after a period of time. Melanoma cells continue to proliferate forever, meaning they are immortalized. They have to sustain the growth of blood vessels. Judah Folkman, who died not very recently, relatively recently, is the one that showed very clearly if you just cut the blood supply to a tumor, the tumor disappears. So the tumor cells essentially maintain angiogenesis by sending all kinds of growth factor and all kinds of agents that signal the blood vessels to grow into the tumor. They have to invade. Okay, they start in the skin. They have to go some other place, as Steve told you, in order to be effective in the remission, which is not very pleasant. And they have to evade the immune response, as Mario said. So what's the mechanism for all that? The mechanism is in different way. We need to have the DNA damage. We have to have genomic instability that change the chromosome and the DNA. And there are a lot of mutations, amplification, and other effects. So going back a little bit to the three major genes that are drivers in melanoma, which I listed them. You have the KIT, which is a receptor. It's a receptor tyrosine kinase. It's all the way up here. This is a receptor that usually responds to growth factor and starts the normal proliferation effect. We have the RAS. So just to go back a little bit, this is outside the cells, this is inside the cells, this is the cell membrane. RAS usually sits in the cell membrane. It's usually activated by the receptor and goes through what we call the MAP kinase cascade and induced proliferation. So all those yellow stars or lights are really the mutation in these three major genes that lie in the same pathway in a way of cell proliferation. So the way we think about it, we inhibit KIT, then we inhibit the whole process. We inhibit BRAF, we inhibit the whole process. Or NRAS, unfortunately, we still don't have any agents that inhibit this protein, although there has been an effort for quite a while. However, that would be very wonderful if we could really eradicate melanoma by hitting BRAF or KIT. Some patients still do not respond, and some relapse. And here you have a figure that I took from one of the publications from Ribas, in which you can see that with time, there is a group of patients that are very poor responders. They still develop, and the tumor progress. And some, not all, some get durable response. So we need to do better. And one of the reasons for this variability in response is that the melanoma is not really a single disease. It's highly diverse, and each tumor is essential, which I'll show you in a minute, is different than the other. And the likelihood of a patient cancer responding can be strongly influenced by the genetic alteration, the other genetic alteration in the tumor. BRAF mutation, what else is there that may overcome the inhibition? So what we have started to do, and what the new concept in cancer genetic, is to find out what are all the other alterations, and especially the mutations in the tumor. So the idea is to take tumor biopsy, to take a tumor, to analyze it by the regular pathological system that we have that determine whether it is a cancer or not, and then to subject the tumor to what is called high sequencing. In this case, I call it MySeq because actually there is new machine that will probably apply one day to the clinic to take individual tumors and sequence them. So these MySeq instruments decide all the mutation and other genomic alterations that are in the tumor. So this will lead to additional classification, not just what was described before, whether it is thin, whether it is invasive, whether it is BRAF or NRAS or KIT mutation. This is now routine in the clinic, but whether there are other changes that may classify this tumor to other specific treatments. So that's a general idea that is now, so to speak, emerging, that at one point will take place. So this particular genetic information will allow us to give actually different treatment to different patients. This is what is called patient-tailored therapy. So the race is on. We and others are really trying to find out what other mutations are in melanoma. And here, I have to really preface the description of what we have done to the organization. This is a very complicated and elaborate endeavor. It requires, and essentially, as already mentioned before, we have the Yale SPOR in skin cancer, which is, the SPOR stands for Special Program of Research Excellence. And we have it for the past 6 years. And this SPOR builds up all the components required to do this kind of sequencing. First of all, we have the community. We have the surgeon, like Steve and Deepak, and the clinician, like Harriet and Mario, that provide us all those tumors and normal DNA, as we see. We have a core in the SPOR that collects all those specimens and track all the clinical information. So by now, I'll show you another slide. We have at least 500 melanoma, and we have matching DNA. Then we are going to accumulate a lot of clinical and mutation information. We need bioinformatics course to make sense out of it, and we have that. We have structure, function expertise, and we have the facility to do the sequencing, which is the care facility. And we have the funding, which is from the NCI, the MRA. Recently, we got some money from Gilead. We have also from patient advocate, from Rose and Jerry Meyer, and the Department of Dermatology. So all this is needed to perform what I just described to you. So the specimen code, this is really very important. And we have right now about 520 melanoma tumors. We have blood. When I say blood, it can be serum and plasma that we collected, and we can do all kinds of immunological studies, and we have normal skins. And the total number of patients that are now in our database is about 1,000. So what exactly is that we are doing? What we did, we took tumor DNA, and blood, and we extracted the DNA. We subjected it to the Illumina sequencer. That's more or less how the machine looks like. And then there is a whole process of determining, one, is there any mutation there? Is what we see different than what is normal in the normal population? Because as it turned out, you might be surprised about it, but each individual in this room has at least 100 to 120 different variants. What I call variants is differences in the DNA that encodes a different protein. Each one of them, not necessarily inherited. This is what is called genetic accumulation of changes that do no harm. They are just there. So we do have to have the normal DNA to compare to the tumor. So the variant calling is really a big bioinformatic core. And then what they do, they essentially compare it to the database and to Yale SNV. The program decides whether it's somatic and inherited, because we are interested also in new inherited genes. We then decide, does it have any effect on the protein or not? You know, having blue eye or brown eye, it just doesn't matter. You know, the eye function as well. We have to know whether the protein is affected in a very deleterious way. And then we decide whether we get novel somatic mutation or whether we discover novel inherited genes. What Danielle, I don't know if she's still here, I don't think so. What she described, really, the inherited genes in melanoma right now are about 2. No more than that. And there must be more than that. So what is that we got? What this plot shows you is the number of mutations. It goes up to 3,500 in different melanomas. So the melanomas are over here, and the number of mutations are here. And what you can see is that we get a group of patients or tumor that have very high mutations from 500. What we consider high is from 500 to about 3,000. And all those samples of tumors are essentially wild type to BRAF. They don't have any BRAF mutation. And they all originated from patients that are older, they are 80 years old, from head and neck, and from chronic sun damage. So this is the kind of mutation that are there for a very long time, accumulate mutation, and then, so to speak, display a malignant phenotype. This is also described here. Here is the number of mutation with age. You can see that with age, the number of mutation in the tumor is increasing. This is the BRAF mutant cells that are really lacking here, and also are not here, except for one of them. This is the group of melanomas that are not in the skin per se. I mean, melanoma are in the skin, but they are mostly coming out of acral, ocular, uveal, and mucosa. All those letters mean O, M, whatever, is essentially what we call non-sun exposed melanoma. So non-sun exposed melanoma have very few mutations. Sun exposed melanoma has intermittent, and the very old have very high mutations. So obviously, what does it mean? Well, what does it mean here? You can see a little bit in this histogram. This is what we call a typical sun damage mutation. This particular change is a sun damage mutation in the DNA. It's a change in the DNA that it's very prevalent in sun-exposed and not present in sun-shielded melanoma. So this is just to enforce the reality that sun is a major, major factor in cutaneous melanoma. And obviously, I'm not going to describe you all the mutations that we discovered, but just the various pathways that they affect. They affect DNA damage. They affect what is called tumor suppressor. Those are the brakes that don't allow the cells to proliferate forever. Mutations that enhance migration and invasion, enhance proliferation, and modulate the immune response. So one of the genes, for example, that we discovered, and I'll finish with that, is a RAS-like gene. So everybody talks about a BRAF and NRAS mutant, as I did until now. Those are 43 in our cohort, 19 in our cohort, and this new one is about 9% of the melanoma have this gene. So in our set of melanoma patients, this is the third most common melanoma gene mutation. And the interesting aspect of this mutation that is prevalent in male, and one of them is sitting right here, it's UHEF. UHEF is talking. You have the mutation in one of these genes, in this gene. Okay, so it's prevalent in male. It's mostly in melanoma from sun-exposed area that reported also excessive sun exposure. The mutation has the UV signature, as I indicated before. It is not inherited, and it confers growth advantage to melanocytes and increase the migration rate of the melanocytes. So why is it mostly in male? In all our cohort, by the way, we have only one female that is about 80 years old that had the melanoma right here in the face with this mutation. In fact, what you can see in this graph is that the number of patients with in head and neck and trunk, well, head and neck is considered sun-exposed area for sure. The number of males is larger in this group compared to females. So what do we think that this gene is doing? We think that this gene is involved in very early malignant process. Here you have the normal skin. Here you have nevus. These cells are the melanocytes. And the melanoma arises first by starting to proliferate. This is your epidermis, the various layers that were discovered before. And what we think that this gene is increase the proliferating rate of the early melanocytes and starts the process of migration. So just to conclude, what is in the future, what we hope that will happen in the future is that sequencing will be part of the clinical care. We hope that every tumor will go an analysis in which the treatment will be determined, either drug resistant or immunotherapy. Once drug resistance actually appears, then another set of analysis, another biopsy will be taken out and will analyze whether maybe a new mutation that arises in the tumor cells with additional mutations started to proliferate that maybe can be addressed or can be inhibited by another drug. And then the process will go on. So each time we'll have new knowledge about the mutation and new knowledge how to treat the patients. And all this work could not have done without a group of physicians and clinicians and basic scientists, as I indicated before. Steve, Deepak, Mario, Harriet. We have Miguel Materin, who is an ocular patient, Marcus, who is a pathologist. And bioinformaticals, you can see the number of people that are taking care of all this sequencing data because they are enormous. We have the structure function analysis. In fact, we crystallized the mutant that I just described to you, have been crystallized, and also the crystal structure indicate what the mutation is doing that can allow also to develop drug against it. We have the Genome Center. We have collaborators in Australia and the funding resources that I indicated before.